Soil is the field production grower's most important physical asset because it is from the soil that the essential mineral elements and water are obtained for plant growth and development. The soil also provides support for the plants and is home for billions of microorganisms. These organisms play a vital role in changing the chemical and physical properties of the soil to make it more fertile and by interacting with roots in both a positive and negative manner. A grower should know the essential mineral elements and how they affect plant growth, their deficiency symptoms, and methods for preventing or correcting deficiencies, the role that soil reaction pH plays in the availability of various mineral elements, and ultimately the growth of the plant, the concept of nutrient element balance and how it affects the growth of plants, how soils and plants are tested for the various elements, and how to interpret the results of the test as they relate to the particular situation. I'm Dr. DeBusk, and in this video, I'll discuss about micro and macronutrients, pH and its importance to the soil, and soil and tissue tests. Although there is no unanimity in defining soil, it is commonly thought of as that portion of the Earth's surface derived from rock and, in some cases, containing organic remains which living organisms are associated. Weathering breaks rocks into smaller particles by the processes of physical disintegration and chemical decomposition. The resulting changed physical and chemical characteristics of the rock make it capable of supporting the growth of plants when supplied with water in the proper ambient atmosphere. The nature of a soil is dependent upon the rocks from which it was derived, but is also dependent upon the length of time and intensity of the chemical, physical, and biological forces that have acted upon it. The soil, in turn, is the source of the essential mineral elements for plant growth. The essential mineral elements, often referred to as plant nutrients, may be divided into two groups. The major or macronutrients include the elements nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. The second group is called the micronutrients because they are needed in very small quantities. They are also known as trace or minor elements. They include boron, copper, iron, manganese, molybdenum, zinc, and chlorine. Nitrogen, N, as a constituent of protein, is involved in all the physiological processes taking place in plants. A deficiency of nitrogen is expressed in woody plants as a decrease in vigor and a foliage color that is light green to yellow, chlorotic. The older leaves express the symptoms first. Under a prolonged deficiency, the leaves scorch and drop off. An excess of nitrogen can result in excessive vegetative growth and decreased flowering and fruiting. An excess of nitrogen may also predispose plants to various pathological and physiological disorders such as fire blight and soft weak growth. Phosphorus, P, is needed for root development, flowering, and fruit formation. It is found in large quantities in meristematic tissue and is essential for cell division. A phosphorus deficiency ultimately results in stunted plants. Early indication of inadequate phosphorus within many species of plants are the development of bronze or purple coloration of the petioles on the underside of the leaves, followed by a yellowing of the leaves, particularly at the time of flowering and fruiting. However, phosphorus deficiency is seldom observed in woody perennial plants growing in the field production nursery or in the landscape. Apparently, the roots which penetrate throughout a vast volume of soil are able to absorb enough phosphorus to supply the plant's needs. Potassium, K, although not an integral part of plant tissue, is important for plants. It modifies the absorption of other nutrient elements, influences the carbohydrate-nitrogen relationship, and is thought to influence the absorption and utilization of water, which in turn affects the drought and possibly the cold temperature resistance of plants. A lack of potassium in broadleaf plants is first explained by a marginal yellowing of the older leaves, followed by scorching and leaf drop. In needle-leaved plants, a deficiency is indicated by an apical brown tip separated by a green basal portion by a yellow band. There is also a decrease in the number of years that the needles persist on the plants. Calcium, Ca, is one of the primary elements in the development of woody plants. It is a constituent of the cell walls, and it modifies the permeability of membranes and is important for development of roots. Because calcium is relatively immobile within the plant, deficiency symptoms are exhibited first in the apical portions. While calcium deficiency is not common in the plants, it is manifested by death of the terminal portions, yellowing, and necrosis of the adjacent young tissues plus severe distortion. Margins are ragged and often rolled. This is more commonly associated with seedlings than with mature plants. Magnesium, Mg, is an integral part of chlorophyll, the green pigments in plants, making it essential for photosynthesis. Also, certain enzyme systems and respiration do not function properly at low magnesium levels. Some form of chlorosis in older leaves is associated with a magnesium deficiency, but the expression varies considerably within the species. It is reported that dead areas developing suddenly between the veins are an indication of inadequate supply of magnesium. In advanced stages of magnesium deficiency, shoots become devoid of leaves except for a few at the tip. Sulfur, S, is a constituent of protein and various volatile compounds. When sulfur is deficient, plants are stunted and the leaves are light green or yellow, starting with the young leaves and progressing to the older leaves. Growth is restricted and stems are thin but remain erect. However, sulfur deficiency has not been a problem in the production of nursery crops. Perhaps this is due to its presence in most complete fertilizers as a contaminant and in various forms of nitrogen, phosphorus, and potassium salts that are used in the production of nursery crops. It is also an air pollutant in industrial areas. Iron, Fe, is a catalyst in chlorophyll formation and for oxidation reduction reactions. Its availability in the soil decreases with high pH, high phosphates, or high concentrations of heavy metals or carbonates. Young leaves exhibit an interveinal chlorosis with the veins remaining green. Woody perennials exhibit dieback of branches if exposed to iron deficiencies for extended periods of time. Manganese, Mn, is needed for assimilation of carbon dioxide and nitrogen metabolism and for the formation of organic acids and keratin. It may be deficient in soils of high pH or if large quantities of calcareous materials have been used as a soil amendment. The deficiency symptoms are similar to those for iron. Early symptoms of manganese toxicity are chlorotic leaves, premature defoliation, and suppressed growth with smooth raised pimples underlined by small dark brown spots in the young bark of some species. In advanced stages, the bark becomes very rough, cracked, and scaly. Manganese toxicity appears to be cultivar specific and is associated with acidic soils, anaerobic conditions, and with some steam pasteurized soils. Zinc, Zn, functions in cell elongation and in seed development. Zinc deficiency, although uncommon in woody ornamentals, produces a yellow to bronze color in older foliage. The leaves are relatively narrow with a waxy margin and rosetted due to reduced internodes. Dieback of shoots can occur where the deficiency is acute. Boron, B, aids in proper root development, flowering, and fruiting and is necessary for nitrogen and carbohydrate metabolism. The application of excess lime may induce boron deficiency, which is more common in sandy soils than in clays. Death of the growing points, brittleness, and dieback of the stems combined with scorched, curled, and often rosetted leaves are symptoms sometimes associated with boron deficiency in field production nurseries. Copper, Cu, is a catalyst in respiration and is needed for carbohydrate and protein metabolism and in seed formation. Although copper deficiency is not too common in woody perennials, it has been reported in young fruit trees and in azalea plants growing for prolonged periods in containers. In fruit trees, deficiency is expressed by a withering and death of young shoots in the late spring or early summer. In azaleas, the new leaves are smaller than normal and soon become scorched. The early symptoms resemble potassium deficiency or frost damage. Molybdenum, Mo, is needed for protein synthesis, but is needed in such minute quantities that it seldom limits growth of woody plants. An initial diagnosis of nutrient deficiencies can often be done through visual symptoms. The chart is broken down into symptoms that first appear in the lower or upper leaves, although symptoms can vary with different plant species, and symptoms can also be caused by other biotic diseases or abiotic disorders. It is recommended to follow up visual symptoms with a soil test or leaf analysis to confirm the diagnosis. The reaction of a soil, pH, is an indication or a measure of the degree of acidity or alkalinity of a soil. It is common to speak of a soil in terms of such as slightly acidic, very acidic, or slightly alkaline. A more accurate method of expressing soil reaction is to express it in terms of the hydrogen ion concentration using the pH scale of 0 to 14. 7 is the neutral point. A pH number lower than 7 indicates acidity, and a number above a 7 indicates alkalinity. The soil reaction has a strong influence upon the availability of the various mineral elements needed for plant growth. Plants grow better when grown in soil at the optimum pH range for the particular species. The macronutrients are readily available within a range of 6.0 to 8.0, but the micronutrients favor more acidic conditions and their availability declines as the pH increases. The most favorable soil reaction for the growth of most woody plants is between a pH of 5.5 and 7.5. Within this range, the essential mineral elements are readily available and the microorganisms of the soil carry on their beneficial functions. However, some species of plants grow best under acid condition, for example gardenias and azaleas, whereas others under more alkaline conditions. Growers should be familiar with the optimum pH range for their crops and select sites accordingly for their production. When the soil reaction is too acid for optimum plant growth, it can be modified by the application of limestone. However, the amount of limestone that needs to be applied will vary with the degree of change that is desired, the soil texture, the content of organic matter, and the form of limestone to be used. When the soil reaction is too alkaline, it can be acidified with the application of sulfur. The amount to be applied will depend upon the soil type and the number of pH units that the soil reaction is to be modified. Best recommendations for the amount of limestone or sulfur to apply are based on soil tests performed by persons trained in the analysis of soils. Affecting a pH change of nursery soils is best done when the land is not in crop production and well in advance of planting the nursery crop. When changing the soil pH more than 1 unit, it may be desirable to make split or multiple applications, testing the soil pH after each application has had a chance to act before applying the next. Various forms of limestone have varying abilities to modify the acidity of soils. The difference in ability is referred to as the neutralizing value and equated on the basis of pure calcium carbonate. For example, the molecular weight of pure calcium hydroxide or hydrated lime is 74. By dividing 100 by 74, the percent value is 135. This indicates that 1 pound of hydrated lime is equal to 1.35 pounds of calcium carbonate. Liming materials react at different rates because of not only their chemical composition, but their coarseness and hardness. The finer limestone is ground, the more quickly it will react. Also, the more porous calcite materials will react more quickly than the harder dolomitic materials. One of the interesting phenomena of nature is the ability of soil to absorb various mineral nutrients from fertilizers and then later to release them to be absorbed by plants for their use in metabolism. The ability of soil to hold certain minerals is known as the cation exchange capacity, or CEC. The exchange capacity of the soil developed as a result of the weathering of rocks into small clay particles, which possess negatively charged crystal surfaces. Clay particles absorb thousands of hydrogen ions, H+, and other exchangeable cations such as calcium, magnesium, potassium, and sodium upon their surfaces. Organic matter can hold both cations and anions such as phosphate, nitrate, and sulfate. They are made available to plants through the decomposition of the organic matter by various microorganisms within the soil. Nutrient element balance is a fundamental concept in plant nutrition. Maximum growth and yield occurs only when optimum nutrient intensity and balance are obtained. An imbalance in elements can result in toxicity or deficiency, which can limit growth. For example, boron toxicity can be induced by a high ratio of calcium plus potassium divided by magnesium, or calcium plus magnesium divided by calcium, but not potassium plus magnesium divided by calcium. A deficiency of manganese can be induced by high accumulation of one or all three bases, calcium, magnesium, and potassium, in proportion to manganese. There may be little or no response to increases in nitrogen fertility unless phosphorus levels are in balance with nitrogen. Heavy phosphorus fertilization of soils can induce deficiencies of iron, copper, manganese, and zinc by combining them to form relatively insoluble compounds. The chemical composition of leaves can be determined by chemical analysis. Because the nutrition of leaves is fairly well stabilized by the end of the growing season, it is the recommended time for collecting samples. For deciduous species and evergreens such as pine, the middle foliage from the current year's growth is suggested. For evergreens such as juniper, the current year's growth is sampled. It may be necessary in some situations to supply the laboratory with healthy foliage for a reference standard, since standards have not been established for all woody species. Although exact fertilizer recommendations cannot be made based solely on leaf analysis, using this data combined with current soil tests, experience with the nursery crop and soils, and information on past fertilizers and cultural practices, a grower can develop a cultural program that will improve the quality of growth. A good soil testing program is necessary for growers to make sound soil management decisions for maintaining adequate pH levels and a reasonable balance of nutrients. Because different extraction techniques are used by soil testing laboratories, it is not always possible to compare test results between laboratories. Soil samples, cores of 12 to 18 inches deep, should be taken at a number of locations in each field to be planted to develop fertilizer recommendations for long-term nursery crops. Most soil testing laboratories will typically provide pH and the availability of macronutrients, as well as lime and fertilizer recommendations for selected crops. You may also be able to get organic matter, micronutrients, and electrical conductivity. Some soil testing laboratories report phosphorus and potassium on the elemental basis. To convert P and K to the oxide form that is found in fertilizer bags, multiply by 2.3 and 1.2 respectively. To convert P2O5 and K2O to the elemental form, multiply by 0.44 and 0.83 respectively. It is very important to select a reliable soil testing laboratory that has experience with the production of nursery crops and to continue to work with the same laboratory. In this soil test example from UF-IFAS, the values of the nutrients are shown as bars. You may notice that nitrogen is not shown, and that is because nitrogen moves too quickly through the soil for the values to matter. So nitrogen recommendations are always provided based on the crop. In this sample, the pH is 5.4, so it is slightly acidic compared to the target pH of 6.0. Also, all of the nutrients are in the very low range, which is very unusual. For field conditions in Florida, the phosphorus levels are often very high. For the nutrients, the amount of the oxide forms are provided so that an appropriate fertilizer can be chosen. The amount of nutrients are based on a yearly amount. For the low pH, a small amount of lime is recommended with a recommendation of dolomitic lime since that form contains magnesium. In conclusion, hopefully you learned about the types of nutrients and their deficiency symptoms, the importance of pH, and testing for nutrients.